Welcome to this lecture on the basics of neuroscience. Today, we are going to explore one of the most fascinating and crucial microscopic structures in the human body, ion channels. If you have ever wondered how your brain processes thoughts, how you feel pain, or how your muscles know when to contract, the answer begins right here. Let us dive in and uncover how these tiny gateways make all of human consciousness and movement possible. To truly understand what these structures are, let us start with a simple analogy. Imagine a highly exclusive nightclub surrounded by a thick, impenetrable brick wall. The inside of this club represents the inside of a brain cell, and the brick wall is the cell membrane. Absolutely nothing can pass through this wall on its own. However, this wall has special, highly regulated doors with strict bouncers standing guard. These doors are what we call ion channels. They do not just let anyone in. They are incredibly picky, allowing only specific guests like sodium or potassium to pass through when the time is right. By opening and closing these doors, the cell completely changes its internal electrical environment. Now that we have a conceptual picture, let us look at the formal biological definition. An ion channel is a specialized transmembrane protein complex that spans the hydrophobic lipid bilayer of a cell membrane. Think of it as a tunnel built out of proteins. These proteins assemble to form a central, water-filled pore that creates a continuous pathway connecting the outside of the cell to the inside. When this pathway is open, it allows ions to rapidly diffuse down their electrochemical gradients. This movement of charged particles is exactly what generates electrical currents across the cell membrane, turning a biological cell into a tiny electrical device. Understanding the structure of these pores brings us to their first major property, which is selectivity. Why does this matter? Because if ions could just flow randomly, the cell would have no control over its electrical state. Selectivity means that the physical shape and the electrical charge of the pore act as a highly specific filter. This filter ensures that only certain inorganic ions can pass through. For example, a sodium channel will only let sodium ions through, while completely ignoring potassium, calcium, or chloride ions. This strict selectivity is what allows the neuron to create very precise electrical signals by moving specific charges at specific times. Beyond just being selective, these channels must also know exactly when to open and close, which brings us to the concept of gating. Gating is the channel's ability to transition between an open state and a closed state. This does not happen randomly. The channels respond to specific triggers in their environment. Some channels open when the overall voltage of the membrane changes, while others open when specific chemical messengers bind to them. This gating mechanism is crucial because it acts as the on and off switch for the electrical currents, giving the brain cell moment-to-moment control over its communication. Before we move on to the mathematics, let us see this gating process visually with a concrete diagram. On the screen, you are looking at a cross-section of the cell membrane. On the left, you can see a closed ion channel. Notice how the orange sodium ions are highly concentrated on the outside, but they can't pass through the blocked pore. On the right, the channel has received a trigger and transitioned to its open state. The pore is now clear, and you can see the orange ions flowing down their gradient into the cell. This simple mechanical change is the physical basis of all neural signaling. Seeing how these channels operate physically leads us to an important question. Why do we care so much about them in medicine? The truth is, ion channels are the fundamental basis of all electrical signaling in the human body. Without them, your neurons could not generate action potentials. In real life, many life-saving drugs target these exact proteins. For instance, when your dentist uses lidocaine to numb your mouth, that drug works by physically plugging up the sodium channels, stopping pain signals from reaching your brain. Furthermore, genetic mutations in these channels cause severe diseases called channelopathies, which can lead to epilepsy or cardiac issues. Now that we understand what ion channels are and why they matter, let us look at how neuroscientists calculate the electrical current flowing through them. This formula is a biological variation of Ohm's law from physics. The term I-ion represents the actual ionic current flowing across the membrane, measured in amperes. This current depends on two things. First is G-ion, the macroscopic conductance, measured in Siemens, which tells us how many channels are currently open. Second is the term in parentheses, Vm minus E-ion. This is the electrochemical driving force. Vm is the actual membrane potential, and E-ion is the equilibrium potential. If conductance increases because more channels open, the current increases. If the driving force is zero, no current flows at all. Let us put this formula to work with a concrete example by calculating the electrical current generated by sodium ions entering a neuron. We are given a sodium conductance of 0.05 millisiemens. The resting membrane potential is negative 65 millivolts, and the equilibrium potential for sodium is 50 millivolts. We need to find the total sodium current. We start by substituting our given values into the ionic current equation. We subtract the equilibrium potential of 50 from the membrane potential of negative 65. This gives us a massive driving force of negative 115 millivolts. Multiplying this by the conductance gives a final answer of negative 5.75 microamperes. This large negative value indicates a very strong inward pull on the sodium ions. Now that we have calculated the sodium current, let us look at what happens with potassium ions in the exact same neuron. This time, the potassium conductance is 0.1 millisiemens. The membrane potential is still negative 65 millivolts, but potassium's equilibrium potential is negative 80 millivolts. We apply the exact same equation. Notice that when we subtract negative 80, it becomes mathematically identical to adding 80. This simplifies our driving force to positive 15 millivolts. Multiplying this gentler driving force by the conductance gives us a final current of positive 1.5 microamperes. The physical meaning of this positive answer is that the combined electrical and chemical forces are pushing potassium in the exact opposite direction compared to sodium. These two examples we just solved bring us to a critical rule about interpreting the direction of ionic currents. In the standard convention of neurophysiology, the sign of the current tells us which way the positive ions are moving. A negative sign, like we saw with sodium, means that positive ions are flowing directly into the cell. We call this an inward current, and it makes the inside of the cell more positive. Conversely, a positive sign, like we saw with potassium, means positive ions are flowing out of the cell. We call this an outward current, which removes positivity from the cell and helps maintain its resting state. We have covered a lot of ground today, so let us summarize the key takeaways from this topic. First, ion channels are essential selective gateways embedded in the membranes of our brain cells. Second, by opening and closing, they act as the physical bridge between the chemical world of ions and the electrical world of nerve impulses. Finally, we can precisely calculate these ionic currents using the driving force and conductance. Understanding these channels is your absolute first step to mastering how neurons communicate and how the brain truly functions. Great job following along.